Hey everyone, welcome back to another MCAT Shemmassian workshop. Today, we're exploring a high-yield Biochem passage pertaining to epigenetic and transcriptional profiling. We'll read the passage paragraph by paragraph, discuss any figures, and then we'll get to all the questions, complete with explanations. Let's get started. Before we jump into paragraph 1, I just want to point out that you can actually find over 4,000 MCAT practice questions on our MCAT question bank. You can find all these questions on our website at shemmassianconsulting.com, but today we're just going to go over a couple of them. Now let's jump into paragraph 1. And paragraph 1 states that triple-negative breast cancer, TNBC, is characterized by poor patient prognosis. TNBC cells lack expression for estrogen receptors, progesterone receptors, and human epidermal growth factor 2 receptors. The human cell line HCC 1806 is used to model the triple-negative breast cancer to study this disease. So, if we were to reveal the highlights, you would see that we would highlight things like cancer. We would also highlight things like lack estrogen receptor, progesterone receptor, and human growth factor receptor. And then the cell line HCC 1806. The reason why we're highlighting these things is because it kind of gives us this idea of like what concepts and what kind of hat should be wearing in this passage. So immediately we're told that this has to do with some type of cancer and that it has to do with a lack of expression for certain types of receptors. What receptor? Estrogen, progesterone, and human epidermal growth factor. We're saying that there's a specific cell line called HCC something, human cell line, and that is used to model this triple-negative breast cancer. So I'm just trying to pick up on all these different words that are simplified into these different letters, right? I don't want to read them as letters. I want to keep it simple. I've got a cancer. That cancer, the cancerous cells lack expression of receptors. What receptors? 3 different types of receptors. And there's a cell line to prove that. Now let's look at paragraph 2. Paragraph 2 states that scientists have recently identified epigenetic dysregulation and chromatin remodeling as potential drivers for triple-negative breast cancer. Researchers hypothesize that a link between the transcription and chromatin structure might provide clues into mechanisms behind the cancer. In the human cell line that's being used to model the cancer, the researchers mapped genome-wide locations for RNA polymerase II, histone variants, and 7 histone modifications. So if I was going to highlight anything, you would see that my highlights would show that dysregulation and chromatin remodeling as a driver for cancer. We would see that there is a possible link between transcription and chromatin structure, and that it was a genome-wide location of RNA polymerase II, histone variants, and histone modifications. And so what this would pretty much show me is that there are 2 things that have to do with drivers for cancer: dysregulation and chromatin remodeling. They're looking at a link between 2 things: transcription and chromatin structure. They're looking at these cell lines, and in these cell lines, they're looking at a genome-wide location, and they're looking for locations of RNA polymerase II, histone variants, and histone modifications. So big things here are like content behind chromatin, as well as transcription, as well as things like RNA polymerase and histones. So that's the content that should be circulating in my head of like what's the content behind these terms and how might those be relevant to the questions. Now let's look at paragraph 3. And paragraph 3 states that the researchers used ProSeq to detect transcriptional responses to a variety of stimuli. ProSeq measures newly synthesized DNA, thereby providing a sensitive tool to detect rapid transcriptional responses instead of measuring only steady-state RNA abundance. In ProSeq, researchers isolate polymerase II, bound to nascent RNA, add biotin to the RNA, digest the polymerase, and sequence the biotinylated RNA. So if we were to reveal the highlights here, there's a couple of things. So we would highlight detect transcriptional responses to a variety of stimuli, measures newly synthesized DNA, It detects transcriptional responses. We would also highlight something like isolate polymerase 2 that's bound to RNA, add biotin to the RNA, digest, and sequence the biotinylated RNA. There's a reason why we highlight those things, and the reason why is because, again, it tells us the methods, right? The flow of the experiment. So we're highlighting things like transcriptional responses, rapid transcriptional responses, and newly synthesized DNA. We're highlighting things like isolating the polymerase, adding the biotin, digesting the polymerase. So we want to walk through the flow of the experiment to kind of get an idea of what happened and possibly why. Now let's look at figure 1. Figure 1 is titled Workflow for ProSeq. And really there's not a ton of information here, and really shouldn't take much long for us to go through this figure. It is just simply the workflow for ProSeq. Things that I'd be paying attention to, or just kind of getting an idea of how this figure relates to the passage explanation of the workflow for ProSeq, right? So we were told the flow of the experiment in the passage, and we can see that there's slightly different information, it's at least described slightly differently, in that, like in the workflow, it shows us nuclei isolation. Didn't say that in the passage specifically, but it does say biotin NTP is being run on with the polymerase 2. So that was talked about. And then it tells us the 3' sequencing. So like there is some slightly new information, or at least a slightly new variation in how this information is being presented compared to the passage. So we just want to be paying attention to those things, again, for the questions. Now let's jump into the final paragraph, paragraph number 4. And paragraph 4 states that after developing the method, researchers used ProSeq to measure the transcription of a putative oncogene after knocking out a related tumor suppressor. So, for to reveal the highlights, we would simply see that transcription of a oncogene after knocking out a tumor suppressor. Those are the things that we'd be highlighting. And again, the reason why is because we're still looking at transcription. We're looking specifically at a oncogene, and it's after knocking out a tumor suppressor. Anytime you get questions on tumor suppressing, or you kind of get it, you usually tend to get questions on whether if you turn something on or turn something off, how would that affect the tumor suppression? Would it lead to more cells? Would it lead to less cells? So they kind of have variations in how they ask that. But anytime you see tumor suppressor or knocking out a tumor suppressor, you want to think about, what does that actually mean? If I knocked out a tumor suppressor, what does that mean? Is that more cells? Is that less cells? How does that mean? What does that mean realistically? So hopefully that passage breakdown felt good so far. We're going to head into the questions now, but before that, we just want to mention again that if you're looking for more information on MCAT buzzwords, please do check out the comprehensive list of high-yield key terms that are listed at the bottom of each content unit in our MCAT Premium Content Hub. We've got a ton of different resources available on our Content Hub, and you can find them all on our website. Now, let's jump into question number 1. Question number 1 is saying that a researcher adds a protease inhibitor to the reaction mixture and finds that polymerase 2 bound RNA can no longer be isolated. It's saying that the protease inhibitor likely does what? And so for this question, it's pretty much saying that you added a protease inhibitor and now polymerase 2 can't be isolated. So what does this inhibitor do? So think about it, right? So ProSeq involves binding RNA polymerase 2 to DNA. Then you add biotin to the RNA, digest the polymerase, and then sequence that RNA. So if you add a protease inhibitor, it stops the protein digestion. So that means that you can't remove the polymerase from the RNA, which means that RNA stays stuck to the protein, which means that it can't be isolated. So because of that, the answer is going to be B, prevents digestion of the protein, thereby not allowing the RNA to be dissociated from the protein. And if we were to look at, like, for example, other options, right? So A, for example, that one would be wrong because it prevents RNA digestion. That's not really relevant because the RNA isn't digested in this step. With answer option C, increased length of sequencing runs, that's unrelated. And with answer option D, prematurely releases RNA, that would actually be the opposite effect. The problem is that the RNA is not released. And so for this question, again, question number 1, our answer is going to be B. Now let's look at question number 2. Question number 2 asks, what is the most likely effect of removing biotin from the experiment? And so you want to be thinking to yourself, where did we talk about biotin? And so biotin was in that flow of the experiment, and it was saying that biotin is added so that the biotinulated RNA can be isolated. And so if you remove biotin, you can't capture or pull down the nascent RNA. So the only answer option that would match for this one would be answer option B, that the RNA can no longer be pulled out of the reaction. If we looked at answer option A, the ones that are wrong, right? So answer option A, protein and RNA dissociate earlier. That would be unrelated to biotin. With answer option C, they bind more tightly. Well, that one's just not true because biotin doesn't affect the binding. With answer option D, DNA degradation. Well, that one also would be unrelated because ProSeq measures transcription, not DNA degradation. So for this one, the only answer option that really matches is the answer option B. Now, let's look at question number 3. Question number 3 is asking, researchers observe a histone post-translational modification in euchromatin. Which of the following do they most likely observe? And so with this question, you want to be thinking about, okay, so what do we know about euchromatin? So euchromatin is like loosely active packed DNA that's associated usually with gene expression. And so if you have active chromatin, that typically has acetylated histones, and that reduces positive charge and loosens the DNA histone binding. So for this one specifically, histone 4 lysine 27 acetylation, that is like a hallmark for transcriptionally active regions. So answer option A would definitely be the best one here. With the other answer options, you want to be thinking of, if it's deacetylation, for example, for answer option B, deacetylation removes acetyl groups. That means tighter chromatin, that means gene silencing. With answer option C, that's wrong. Arginine 4-methylation. Well, one, that's the incorrect residue, and methylation can either activate or repress depending on its site. So that isn't typical for euchromatin activation. And with arginine 8-methylation, that also is associated with usually heterochromatin, which is usually repression. So again, for question number 3, answer option A would be the best match. Now let's look at question number 4. Question number 4 asks, what is the most likely ProSeq result from the experiment the researchers conduct in the final paragraph? So what's the result from that final paragraph? And so remember when I was reading that paragraph, I was saying a lot of the times they ask you these questions about whether something's actually suppressing or it's supposed to increase or change or more cells or less cells. So this is pretty much one of those questions. And so you want to be thinking, tumor suppressor genes usually inhibit oncogenes. And if you were to knock out a tumor suppressor, that means you remove a tumor suppressor. So you have a loss of inhibition. And if you have a loss of inhibition, that means that the oncogene transcription increases. So for this one, the only logical answer option would be answer option C, that the oncogene transcription increases. The other ones would be wrong because A, the tumor suppressor protein expression increases. That one just doesn't make sense because you just knocked it out. With answer option B, tumor suppressor expression is unchanged. That one's just not correct because something changed. And with answer option D, oncogene transcription decreases. That would be the exact opposite of what you would expect. So for this one, question number 4, answer option C is going to be correct. Now let's go into the final question, question number 5. Question number 5 asks that researchers add estrogen to a culture of the human cell line cells. A control culture of those cells receives DMSO. ProSeq data most likely indicates what? And so with this question, you want to be thinking. So those cancer cells lack different types of receptors, remember? So they lack estrogen receptors. So adding estrogen won't activate estrogen-related transcription pathways. So transcription profile should look similar between treated and controlled cells. So the only answer option that would match here would be answer option B, similar new gene transcription profiles for the two cultures. And like the way that you would answer this is like you would be thinking, okay, where do we talk about these cells and where did we get information about it? And that was in that first paragraph where it talked about, what receptors are lacking and things like that. So our answer option is B, but let's go over the ones that were wrong. So like answer option A, increase in estrogen signaling. Well, that one can't happen because there's no receptors. The answer option C, upregulation of estrogen receptor. ProSeq measures transcription output, not the creation of receptors, the receptor proteins. So that one would be wrong. And with answer option D, downregulation, that one wouldn't make sense because those are irrelevant because there's no estrogen receptors expression to begin with. So our final answer for question number 5 is going to be B. And with that, it looks like we survived to the end of a fun biochem passage. So these passages are always a ton of fun, aren't they? Thanks for joining, and we hope this walkthrough was helpful in your MCAT journey. If you're looking for more practice resources, you can always visit our MCAT question bank or premium content hub for thousands of sample problems. Or if you want a more personalized approach, our MCAT experts provide one-on-one tutoring, as well as individualized study schedules and test-taking strategies that go a step further than just content review. Check out our website to learn more, or feel free to drop a comment here with any questions you may have. Happy studying.